Hi, everyone. It's nice to be back at Duke. I'm thank you to the organizers for this chance to speak, and I'm going to talk about fast 3D microscopy to capture the dynamics of life. So my lab at Columbia has really been dedicated to developing all kinds of different optical techniques to look at living tissues. And I've worked on a lot of different scales, from originally way back doing diffuse optics to doing microscopy, some small animal molecular imaging. And in general, the theme has been just to try to go really, really fast and to measure as many things as I possibly could, if possible, in 3 dimensions. And that's sort of rational if you look at the massive explosion in beautiful tools, so many of which have come from Ed, that allow us to sort of label and read out this rich functional information from these living tissues. And of course, because living tissues are generally 3-dimensional, fast moving, and if you have to cut them up to image them, they don't do stuff anymore. So so my lab's worked in a lot of different types of tissue, but more recently, had a stronger focus on brain. I have a whole other half of my lab. I'm not going to really talk too much about this today, but we also put a lot of work into studying neural dynamics, real-time neural dynamics, and that all started with trying to understand the relationship between neural activity and blood flow. So to try to kind of chart the kind of imaging results that we get at the cellular level to the sorts of things that we can do in humans using functional MRI. So this is a whole other area called neurovascular coupling that I also do a lot of work in. And this is a great thing to study when you develop high-speed 3D multimodal microscopy methods, because it's really hard to study if you don't have those. Okay, but so let's do some very basic stuff. How do we usually image in 3 dimensions? Probably most of you know that you're supposed to use a 2-photon microscope to image in a mouse's brain. It's an amazing technology that we've had for about 30 years now. And to collect an image like this one on the left here, which we collected in a living mouse showing astrocytes and blood vessels, you focus a high numerical aperture point and you scan it around in 3 dimensions. You read out the signal from each of those locations, one after the other, and you're able to form this beautiful 3-dimensional image. Now, we can image down to about 500 microns without too much trouble. Some 3-photon methods now can get you down over 1 millimeter, but this whole volume took us about 5 minutes to collect. Now, what we want to actually be able to do is image this entire volume so fast that we could, for example, see neurons firing all throughout that region. So you can do some simple math here. You can say, if I'm going to image 400 by 400 by 100 pixels, that's 16 million points. Okay, your Ti-Sapphire laser in your 2-photon microscope pulses at 80 megahertz. So if you go at 1 pulse per pixel, you can never image this volume any faster than 5 volumes per second, which is not fast enough to pick up the activity of all of those neurons. Plus, if you were just doing confocal, you would now be looking at each pixel for only 12.5 nanoseconds, which, if you compare it to GFP lifetime, is 2.4 nanoseconds. So you're basically right at the limit of even being able to interact with your fluorophore. And you would be scanning your spot at 200,000 lines per second, about 10 times faster than your average resonance scanner. So that didn't stop us from actually trying to build a system like this until we did this math, and we said, no matter what we do, we're never going to be able to get past this limit of 3D imaging if we're just going to scan one spot around one after the other. So around the time I was starting my lab, I went to an amazing talk on, back then, something called ultramicroscopy, and got very interested in Lightsheet microscopy, which was forming images in a way that didn't seem to match up with all the things I'd been taught. So in Lightsheet, and back then it was done mostly in, say, cleared tissues, what I learned was that it's a technique where you can create a sheet of light. You can and then focus your camera onto that sheet, and that actually gives you optical sectioning, because your point spread function is then the product of your excitation light sheet point spread function and your detection cone. So you can get diffraction limited 3-dimensional imaging forming your image this way instead of by scanning a spot around. Now, if you ask people what are the advantages of light sheet microscopy, you'll hear that it's low photobleaching. And this is shown here by Tim Hawley's lab, who used their light sheet method to image the same sample for 20,000 times, and they still had reasonable amount of fluorescence left. When they did the same sample on confocal, all of the fluorescence was gone within about 200 frames. But the question is why? Why is light sheet so much better? What you'll hear is it's called selective plane illumination. And that means that you're just illuminating the plane that you're imaging at a given moment in time. If you're doing confocal microscopy or spinning disk, you're putting your light in from above, which means it has to go through the tissue above the plane of interest, and then it all gets dumped into the tissue below your plane of interest. So every additional plane that you collect in your three-dimensional image is like re-imaging every plane over and over and over again. In Lightsheet, you can just illuminate the plane that you're actually looking at very selectively, and therefore you don't illuminate the tissue above and below, and you save it from all of that horrible phototoxic light. But another real benefit comes from this point scanning thing. So if I'm actually illuminating a plane and then I'm using a camera to image that plane, and we're talking about a 400 by 400 pixel plane, my camera is going to have 160,000 times longer to integrate signal across each of those pixels because it's doing so for all of them in parallel. And so this can allow us then to use cameras because we're in a much better regime for noise equivalent power. And plus that means we can use much lower levels of light for longer as opposed to really, really high intense levels of light for a short period of time, which apparently things like fluorescent proteins like much better. The other really nice thing about Lightsheet, which took us a while to figure out, was that we are also giving the photons multiple chances to interact along the sheet. So if you think about doing high numerical aperture confocal, you're sending your photons in, but any of them that have an interaction above and below your plane of interest, you block, you try to get rid of using your pinhole. So they really only have one single chance to have a meaningful interaction. Whereas when you're sending your light down either in an axial beam or in a sheet, if it doesn't interact in the beginning, it carries on. It has another chance to interact and another chance. So basically every fluorophore, every fluorescent photon that's generated in the tissue is going to actually count and be useful to you, as opposed to in these other cases where you throw all of those away. And so what that means is that you're going to get more emission photons per milliwatt of light that you put in. So all of those things actually come together, and we've done some modeling on this, taking into account the nonlinearity of photobleaching of fluorescent proteins. And so what we can see is that it's not just that it may be more practical to use Lightsheet for high-speed 3D imaging, but that there's this massive advantage in terms of then your signal to noise and your efficiency of your photons. If you were going to image at 10 volumes per second doing point scanning, you would literally have no light left at all after 10 seconds if you were doing 10 volumes per second. But using Lightsheet, you'd have plenty of light left, which you can leverage to get yourself the kinds of signal to noise that you need for very high-speed imaging. So if Lightsheet has this amazing potential to do diffraction limited imaging as well as very fast imaging in 3 dimensions, why are we not using it all the time for everything? So the big problem here is the geometry of Lightsheet microscopy. So as I mentioned, you generally bring the light in from the side, you focus onto that plane, and then in order to create 3-dimensional image, you actually have to move those 2 planes up and down in synchrony with each other, which can be quite challenging to do quickly. Or you have to physically move your sample up and down, which means you have to hold on to your sample in some way and have it fixed or mounted. And so when you have all of these different issues, you've got these objective lenses around here as well. You've got working distance limitations, immersion medium limitations, and you have to learn how to mount your samples into these little cylindrical tubes. This is fantastic for things like embryogenesis, but it's not going to be useful for generalizable use in the way you would normally use like a confocal. So we came up with this idea instead of illuminating the tissue using an oblique plane of light coming out of our primary objective lens and then detecting the light back through the same objective lens. Now, at high numerical aperture, this gives us about a 90-degree crossing angle between our excitation and emission, so we still get our nice isotropic resolution, but you now see that it completely removes this need to have the sample hovering in free space. So you can use water immersion here, high numerical aperture objective lenses, and your system that you're imaging can be completely intact. It can be a sample on a slide, it can be a brain, a tongue, it can be anything. And just to be clear, what I'm going to explain is that we are getting our camera focal plane to be on this oblique plane. And it's very common for people to tell me that this won't work and I can't do it, but I will explain to you in a minute that we can. And I'll say that, you know, this idea of doing, it's essentially remote focusing, it's mapping the object plane to an intermediate image plane. You can get this perfect 3-dimensional mapping, which allows you to then use objective lenses to create these kinds of oblique images and move them around without actually breaking the laws of physics. So the thing that we really added to this was the ability to turn this weird imaging geometry into a 3-dimensional microscope. Now, we could have moved the objective up and down or from side to side or moved the sample from side to side, but we came up with a way to just actually scan the sheet from side to side without having any other moving parts. And I'll show you that real quick. If I bring my light in here, it's a low numerical aperture beam on the edge of my system. It's going to hit the back of the objective lens here, and it's going to come out on an angle. And if I have a cylindrical lens or other beam conditioning optics back here, this is going to come out as a plane of light into my sample as I want it. And if I look at light that's coming back from the sample, it's going to form an intermediate image over here using that perfect 3D mapping because of these matched two arms of this telescope. It's going to create an intermediate image of that oblique plane over here. If I want to image that plane, then I can just focus my camera onto that plane. And that then is like having the camera actually down here focused on that plane. So we've now created this image, light sheet imaging condition. So then what we do is we have a galvo mirror in the system, and that allows us to move the galvo mirror from side to side, which causes the sheet to scan from side to side in the way that I want it. But the sort of magic of SCAPE is that when it's in this moved position and the sheet is moved to the side, you can see that the light coming back, just like in a confocal microscope, has all its ray directions corrected. And so this sheet over here stays stationary and stays beautifully focused onto the camera. So I can move this galvo mirror from side to side however I want it. It's going to move the sheet from side to side, but I'm going to get the equivalent of moving my excitation sheet and my detection plane in parallel at the same time, however I want. So I can put my sample under my objective lens here, in this case, a little zebrafish in a drop of agar. I can image it from above like this. And if I want to go at 20 volumes per second, I'm literally moving my galvo at 20 lines per second, which means this is a very cheap, simple, easy galvo. And this is just the only moving part in the system. Down here, you'll see images of the zebrafish here that would be coming out of the system, only I've slowed them down massively because otherwise they'd be too fast to see. And we routinely use image splitters in front of our cameras, which project two different colors of image side by side. This means that we don't need to read out more rows of the camera, but we can collect two colors at the same time. And incidentally, because each image that we collect is an oblique plane, we're getting basically the depth into our sample is one of our dimensions of our camera image. So it's not uncommon for us to only want to do 100 or 200 depths into a sample. You can read out a standard SCMOS camera at over 2,000 frames per second when you're only reading out 100 rows. So we leverage that speed to be able to then sample laterally across the sample to get to over 10 volume per second imaging. And all I do with those oblique planes is just literally stack them back together, and then I have an optically sectioned three-dimensional image. So it's really important here that we don't actually use any reconstruction algorithms. This system, the basic system generates 1.4 terabytes of data an hour. So these data sets are really huge, and anything that you have to do on each image to fix it, to correct it, to deconvolve it is almost crippling. So having just pure data coming right out that you can look at immediately, especially for things like in vivo experiments, is really important. So the system is also remarkably sort of inexpensive and simple. This is a system that we took in a suitcase to Dresden a few years ago to a course, and we're able to put it back together again and do demos for 2 weeks. You can see it just has a little laser in the back here, galvo here. Then this is our image splitter, that weird image rotation, and the camera here. And one nice thing about having no fuss around your primary objective lens and no piezos or anything is that you can actually spin it around. It works at any angle, which was very popular with people because they could image their sample, you know, in any orientation they wanted. And here's a little movie. I hope my videos are coming through okay. Of the fish here being imaged. Those are the planes of coming out. The fish is just sitting here in a dish under this 20x objective lens. This is just a 1.0 water immersion, 2mm working distance objective. And that's Venkata, who put his heart and soul into developing this version of SCAPE for his PhD. And what we were imaging here was epileptic seizures in zebrafish, again, here generating 1.5 terabytes an hour. Okay, so I'll see if this will run. It's probably clunky. I would love to spend the next 40 minutes showing you everything I've ever imaged with SCAPE. I'm just going to leave this up for a moment. I know Joe knows this about me. So I'm just showing you here a whole range of different applications that we have developed here. And we really, the whole lab had to change what it was doing to accommodate all the people that sort of came and knocked on the door and said that, you know, they absolutely had to have our help to image all of these different things. But it's been an amazing journey for us to actually, you know, learn about all of these new organisms and actually learn new science, as well as, you know, getting this into the hands of a lot of new people. And a lot of these new specimens that we image then inspire us to develop new things or add capabilities to the system. Okay, but still people ask me, why do we need fast 3D imaging? So I'll just go through it really quick. So firstly, it's because living things are usually three-dimensional and cellular processes are fast. So here, this is actually a sped up movie of this fish having these spontaneous epileptic seizures. And what we're imaging here is called GCAMP. It's a calcium sensitive green fluorescent protein. So you can express this in neurons and it nicely becomes brighter as the neurons are firing an activity. And there's been an explosion in these kinds of probes now, again, mostly driven by Ed Boyden and people like Ed, not that there's many people like Ed, who can now look at voltage and pH and glucose and you name it. So these kinds of dynamic optical readouts of function are really coming along. And so what you can do when you're actually imaging the whole volume in 3 dimensions all at once is this kind of analysis where then you can go back and say exactly where did this seizure start? How did it spread? Was it the same every time? You could give the animal a drug. You can look at how that impacts the way that the seizures are occurring. None of this you would be able to do if you were doing kind of sequential building up of your 3-dimensional volume using 2-dimensional planes. In another recent study, we worked with this preparation, which is the olfactory epithelium in the mouse brain. So this is an in vitro hemihead living mouse prep. We expressed that calcium indicator GCAMP in these olfactory sensory neurons, and we were able to image them intact inside of the olfactory turbinates of the nose and extract out individual single cell responses as we put odors over the top of the tissue. And each of these cells expresses its own a single G protein coupled receptor, which is sensitive to a specific odor. And that is how your brain actually knows the things that you're sniffing. It's in the code of the different neurons that actually get excited by each specific odor. What we were able to do here was actually apply a whole sequence of not just odors one after the other, but mixtures of different odors, which allowed us to characterize the way in which each individual cell responded, not just to a single odor, but to different combinations of odors. And what we found was that there was this very weird extra little combinatorial thing that caused some of the cells to actually either get inhibited or overstimulated by odors in the presence of another odor, even if you didn't actually smell that odor, even if that odor didn't itself evoke a response in the neuron. And so we worked really hard on this. This is something we published in Science last year. Okay, why else? Because living things move around. So this is a beating heart of a zebrafish. It's imaged at 100 volumes per second now. This is in 2 colors as well, so we can look at individual red blood cells moving through the heart. Again, just purely measured in 3 dimensions without any kind of reconstruction. I've actually slowed the video down here so that you can see the dynamics. And so we can do quantitative analysis on this. And the idea here is for us to be able to really phenotype these zebrafish hearts, looking at congenital abnormalities and the way in which they develop, not just now looking at how they structurally develop, but how they functionally develop for those different abnormalities. And of course, things that move signal. And so this is a combination of those 2 things where we're expressing calcium indicator in the cardiac wall, which is allowing us to not just read out the movement of the wall, but also the signaling that's causing it to move. And so here we worked with Kamara Katoro in Japan, who developed a deep learning algorithm to track the 3-dimensional spots on this heart wall and allowed us to look at single cell calcium dynamics actually in a freely beating heart. And here's just some numbers for you physics nerds. So in collecting this, we're using an intensified camera. We were actually imaging at over 12,000 frames per second to form this 3-dimensional image at 100 volumes per second, but we were still only using about 1 milliwatt of 488 nanometer excitation of the sample. In another study with my collaborator Wes Gruber, he was very interested in the fruit fly larva, and we wanted to know how exactly neurons on the fruit fly larva's body wall were telling the fruit fly larva where it was. And so we were able to just let these larvae just sit in a little channel and crawl as we did high-speed 3D imaging. So here's imaging at 10 volumes per second. There's a top-down projection at the top and a side projection. And so we can see these dendrites of these proprioceptive neurons being kind of crushed and compressed as the larva crawls. By then co-expressing GCAMP in the different channel, we can now look at the way in which not just the dendrites are being moved, but how they're signaling in response to the way that they're being moved. And we developed a lot of tracking algorithms to extract all of this information and characterize the way in which all different types of these neurons actually are representing the movement and the behavior of the animal. even allowing us to develop a geometric model of the larva where we can actually predict exactly where its head was based only on the activity patterns that we were seeing in these neurons. And then finally, the other reason why we need high-speed 3D microscopy is that apparently microscopy is really boring. There is a stock photo from microscopy boredom that you can find on the internet, and that makes me very, very sad. So it turns out that a lot of people spend a lot of time sitting in core facilities trying to take stacks of their samples. This is a sample actually from Ed's lab. Now, as you heard, expansion microscopy is amazing. It allows you to get this incredible resolution, but the samples become really, really big, which means they're really, really slow to image. So this 4 millimeter long strip of expanded tissue took us just 8 seconds to image in our system. So we've been doing a lot of work now exploring how to leverage the capabilities of our system for very high content, also high throughput structural imaging of tissues to kind of get past the current bottleneck that's being faced by all of these beautiful technologies that Ed and his colleagues are developing. We also found that we can image very nicely inside of things like these kinds of microfluidic chips, or PDMS chips. And so we don't have to destroy or open these in order to get to the tissues within them and image them. And again, we had a lot of people interested in looking at these in terms of looking at cellular development and growth and movement. But now we've unlocked a lot of ability to do things like looking at flow and transcytosis, calcium signaling, the response of tissues to drugs, and other kinds of perturbations. And then finally, I just want to show you a little bit of our sort of most recent work where we decided to go back to that very original question that we had about whether or not we could leverage this for imaging the mouse brain. And so we, as I said, point scanning is just simply not fast enough. So we wanted to ask the question, can we do 1 photon imaging of the mouse brain, or actually can we also make a 2 photon version of SCAPE? So indeed, we can do 1 photon imaging of the mouse brain. So this is a really nice data set that we got, imaged at 10 volumes per second with a red dye injected into a Tie2 GFP mouse with its endothelial cells labeled. And as we flick the animal's whisker here, we can read out from all these little squares, all of this 3-dimensional information about the way that this vascular tree is responding just to this one single stimulus event. So this is like real-time imaging of a 3-dimensional vascular network. And one thing we found here that was really interesting is that we never saw the network respond the same way twice. So studies that are trying to kind of understand this network response by imaging little segments of the whole vascular tree and not necessarily picking up a full representation of what it does. We can also image neural activity. So here, this is in collaboration with Randy Bruno, looking at apical dendrites, again, at 10 volumes per second, imaged in the mouse brain with 488 nanometer excitation. And here we've got 150 megahertz pixel rate, which is nearly twice what you could ever hope to achieve doing point scanning 2 photon. And we can apply dimensionality reduction methods. This is a spatiotemporal unmixing strategy that I've been very fond of for years, back in the day, applying it to mice, and now applying it to neural data. So we can basically take that lightning strike movie and turn it into this very nice color-coded movie that shows us each individual neuron separated based on its own unique time course. And we did indeed manage to build 2 photon SCAPE. So here's some data that we collected at 6 volumes per second in the brain, looking at layer 2-3 neurons, and again, striving to get really isotropic 3-dimensional imaging, not just multi-plane imaging. And again, applying those spatiotemporal mixing methods, we can do really cool things here, like actually pull out the signals of firing patterns all throughout the 3-dimensional cell in real time, as opposed to just doing single plane analysis there. And real quick, I don't want to overstay. I'm probably already being warned I have to stop speaking. So here we go. Yep. All right, I'll do this last one super quick. Oh, sorry. Well, and we've also been using this in fish. Okay, so finally, we got very interested in whether we could translate this technology into imaging people, where I'm sure this crowd is very aware of the challenge of intrasurgical guidance, trying to understand, in this case, what robotic surgery is looking at, and this need still to have to take tissues out of the body, preserve them, cut them up, slice them up, stain them, and look at them, and then get back to the surgeon later and tell them what they were looking at in this surgical field. Now we have confocal microendoscopy methods, but they're typically quite slow. They can do a 2-dimensional field of view that's reasonably small, and they generally require intravenous contrast to give enough signal to be able to actually see what's going on. The creeper in my lab, who is graduating her PhD tomorrow, took some tissues and put them under SCAPE and found these beautiful structures that we could see, almost all actually only using autofluorescence. And so this is a living mouse kidney in vivo. Oh no, and the video is not found. Well, there you go. That's okay. So we're working with Shana Coley, who is a pathologist who is a kidney expert, and this is a piece of human kidney now that we were able to get completely fresh, completely unstained, imaging it using SCAPE, finding these amazing structures which match up very nicely to histopathology. And each of our images isn't just able to be collected in situ, in vivo, using intrinsic fluorescence instantaneously, but also each of the images we get is three-dimensional. So we're very keen on developing this, miniaturizing this, and getting this into clinical use. And then finally, we've worked really hard to share the system, take it to courses, show people how to use it, show people how to build their own systems, as well as working with Leica Microsystems to commercialize it. With that, I just want to thank my students, my postdocs and my staff and everybody in the lab. This is my SCAPE team currently. These are my alums. Matt and Venkata put a lot of the early work into SCAPE. And as I said, I didn't really even mention the brain physiology work that we do, but I have another team as well that works on looking at brain-wide activity and blood flow and physiology. And with that, I'll just acknowledge my funding and thank you.